So hi everyone, so I'm Emmanuelle Cameau, and I'm really happy to be here today with you. And yeah, I have put together some thoughts of how we can work on driving the cost down to get all these therapies more available, and it's really food for thought, let's say. Hopefully, yeah. So we will first go take a look a bit on the cost of those challenges that I call for the gene therapy products, how we can reduce cost of those through process optimization, how we can also leverage cost modeling tools to understand the process cost drivers and the impact of process optimization with a case study, and also how process intensification can be used to optimize the cost. So I think if you're here, you know this, but it's really since a few years a golden time for genomic medicines. We have an increasing number of approved products, and we had 6 in 2021. I think we are aiming to more than 10 for 2022. We have more than 200 clinical trials in phase 3, increasing investments driven by venture capital, and also more than 2,000 clinical trials investigating cell and gene therapies globally, and a lot of money raised by gene therapy, advanced therapy companies, so more than 23 billion. As I said, we still have many regulatory approvals that are pending this year. This is an overview of the approvals that are there. The pending approvals are listed in table, and in blue it's all the BLAs that have already been submitted. And so by 2025, we could expect between 10 to 20 approvals. So it's really increasing every year. So when we look at the cost, so of course, viral vector gene therapies, they bring a lot of possibilities of treatment, but traditionally it comes with a high cost of those. This is illustrated by the examples below, which are Luxturna, which this one is the first AAV gene therapy to provide to improve the functional vision in patients that have an inherited retinal disease, and the therapy is around $430,000 per eye, which is 850 total, if the twice are concerned, of course. And then, of course, Zolgensma, that I'm sure many of you are familiar with, which is an AAV-based gene therapy for treatment of spinal muscular atrophy. And this one is a $2.1 million therapy, which is literally the most expensive drug on the market. And the reason for this is mostly a lack of process maturity and adapted technologies to manufacture these products. So let's take a look a bit more in depth there. So the industry is quickly evolving by looking at how to bring innovation in the way we produce. So the technology providers really have a key role to play in this work by developing better technologies, better tools to manufacture these products while reducing the cost. And this cannot be done without the very good collaboration between the manufacturers and the suppliers. So this is really very, very important. I mean, as a supplier, we do it ourselves, but it is really key to the overall improvement and the objective, which is the cost. So if we look a bit more on the process side of things, On the upstream, we can improve, optimize the process through cell line optimization, process optimization, so really looking at the overall process. We can optimize the plasmids, and we can also develop better stable cell lines, for example. On the downstream, it's mostly process optimization, better recovery of the product, and of course, across these two, it's also more adapted technologies, because right up to now, what we have is that we are manufacturing vectors with technologies that have been developed for maps. So there is also maybe something there that needs to be changed to have technologies that are more appropriate to these products. But when we look at these two, in downstream, the average yield for an AAV downstream process is around 25%. So if we're able to go to 100, which is highly unlikely, it would be already great if we will be able to reach a MAB-like yield, which 70–80%, we would only increase the productivity by 4, or the total recovery, which would only decrease the cost by 4. So the key to really decreasing the cost is, yes, we can work on the downstream, but the real big lever to drive the cost massively, which when you talk to some people, it's hopefully between 10 and 100 times, is the upstream side. That is where we, the industry, needs to focus and work to be able to finally drive the costs down. So if we have an increased upstream productivity, there are different benefits that would directly fall from that. So the first one is you would get more doses per batch, which means you have more patients treated, and you then have a reduced cost per dose. You also might have less batches required, which means less material needed to manufacture your product and less labor. And then you also might have through increased downstream productivity, through optimization of the upstream side, a bioreactor size reduction, or less bioreactor skid required, which means less consumable costs, less capex investment, and a reduced footprint overall, which means a smaller factory. So all of those translate directly into a cost reduction. So this really highlights the high importance of process development and optimization, because maybe at small scale, it doesn't seem much, but when you scale up to the manufacturing scale in a manufacturing setup with X number of batches per year, then it all means something. I mean, really, it can mean a lot. So an example, I will give you an example of what I'm trying to say with a case study I did with a cost modeling tool a couple of years ago for AAV scale up. And during this work, we look at technology selection for the upstream side. And when we look at selecting the manufacturing technology, there's different things we need to consider. Of course, first, if we think adherent or suspension, it will depend what is the status of the cell line that is used. But it is also a balance between the scalability, the quality, and the cost. So it's important to understand what are the main cost drivers of the process, what are the different factors that will affect the scalability, and how does each unit operation affect the functionality and quality of the other ones around it? Because when you might change something in one unit operation, and we'll also go a bit deeper in a later slide, you need to think of how will that impact the rest. Cost modeling gives a comprehensive view of the footprint and capital investment you will need, the process duration and manpower required. It can calculate all the media, buffer, reagents needed, and also all the consumables. So in this work that I outline here that has been published, there's a comparison for an AAV process between a multi-tray process, a suspension bioreactor process, and a fixed bed isolate bioreactor process with a common downstream. So they all have the same downstream. There's really a focus on the comparison of the upstream side. So what is really important to have in mind when we talk about cost modeling is that we need to make a certain number of assumptions. And as soon as one of those assumptions is not true, the output can change. And so what I'm going to show you right after these on the outputs of the model and the conclusions you can take out of it is only valid with these assumptions. Okay, but it gives a general idea, a general trend. But of course, if in your case some of the assumptions or parameters are very, very different than these assumptions, then the output might be significantly different than this. Okay, I just want to make clear that this is clear for everyone. Sorry. So we've made assumptions on transfection cell densities, on prices as well, of course, of all the medias, consumables. We assumed a certain PI-DNA ratio, and we assumed a certain titer in upstream as well. And then for all the downstream, as I said, we have a common downstream for all, where we have an assumed yield, an assumed binding capacity, and an assumed dose as well for the therapy, and an assumed batch cost for QC. What we have varied is, as you can see here on the top, we have a multi-tray process, but then we have on the suspension and the fixed bed bioreactor, we have normal and we have optimized. The difference between these two is that we have, for the optimized one, the DNA per cell is decreased at the transfection, and the transfection cell density is a bit higher for both. So for the optimized setup. So this is just for you to take in mind, because we are going to see the output of that. So for this modeling, I used Biosolve software. So this is a quite nice tool. I know there are others out there. You can also build your own. It is certainly quite complex to use. You need to be well trained, but it is really insightful. So it will generate a comprehensive view of a factory that will include the footprint required and the capital investment as well, the process duration and workload, all the use of reagents, and also the consumables. So if we look at the outputs, of first around the cost of scaling out versus scaling up. we clearly see that for the, if we compare here the multi-tray process and the, for example, the bioreactor process, let's not compare them, it's very similar. Whether it is suspension or adherent fixed bed, we see that when we scale, so when we scale up, bioreactors reduce the OPEX by roughly 50%. And the multi-tray in upstream really will increases the OPEX. So this is two statements are linked. If we look at the footprint, which is the graph below, we also see that the footprint, which is here in gray, in light gray, is reduced by roughly 2 times. Sorry, it is, if you stay with multi-tray, between your clinical and manufacturing scale, the footprint increases here. So from this one in 200-liter multi-tray, we go to a higher footprint here, so roughly the double. While with a bioreactor, whether it is suspension or adherent, the footprint stays the same. It means you can do your clinical and manufacturing scale, facility, production in the same factory. You don't need to expand. So this is when we look at the multi-tray versus the bioreactors. And then if we look between fixed bed and suspension, they have, as I said, they have similar capex, opex, and footprint. There is a potentially, in this setup, with these assumptions, potentially small cost reduction of those. So it's the pink here with the fixed bed bioreactor compared to suspension, but it's very, very light. It's 4%. So I would say it's highly similar. And we have, there's a smaller throughput here. Sorry, it was the blue. But in the pink, there's a slightly smaller throughput in the fixed bed, because with the assumptions how they have been modeled here and the process how it has been set up, the fixed bed bioreactor is seated at a smaller cell density, because it's a technology that can let you do that, and therefore the hardware immobilization is longer. So mathematically, you can do less batches per year. So the optimization of in the fixed bed bioreactor, the process optimization allows a small cost of goods reduction. So here, compared to the optimized bioreactor, but it's very, very small. And and this is also why I mean, I have talked about this previously in other venues, but the choice of technology, if this is something we are wondering, really needs to be based on process development risks. What is your cell line? What are what is your target scale? A lot of different things, but certainly the cost is highly comparable between the two. So when we look at the cutout, so how the cost splits between the different components of the process of the cost, we see that there's significant impact of labor and USP consumables. So USP consumables are in orange here, and labor is in blue, dark blue here. So of course, the USP consumables, this is highly valid for the multi-trays and the labor. So you see, this is almost half of the cost, or let's say 1/3 of the cost. is only consumables and labor for the multi-tray setup. This decreases dramatically for the optimized, for the suspension and the isolate bioreactor. It goes down to roughly between 10 and 15% cumulative. What we see, though, is that the transfection mix, so the pDNA and PEI combined, is a common cost driver across all processes, but because of the process optimization that is possible in the isolate, where in theory we can decrease further the quantity of DNA per cell, we can reduce by 50% here compared to the suspension. But again, this is an assumption. It is possible that through increased process optimization, we can reduce that further in the suspension, but it's depending what we can do today. So then I said, yeah, and as I said, the number, if we look here, here we have the batch cost across the different scales. So you see the big difference in batch cost here in numbers. So previously we had it on a bar, and the dose cost. And so the dose cost decreases from between, let's say, 1/3 to 1/2, depending on the process, compared to the benchmark, which is the multi-tray at 1,000 liter scale. So DNA and PEI being this big chunk of the cost, it is really, really important that whatever the platform you choose to manufacture a vector, whether it is traditional 2D, a bioreactor, suspension, or adherent, you need to optimize the pDNA use. So this needs to be a high focus during process development to make sure that once you are at large scale, the cost is well controlled. So this work shows that cost modeling can give additional insight into the technology selection, as I said. We see that bioreactors, they have a significant cost and footprint advantage over multi-tray stacks. And also of course, this is applicable. These are transient processes. In this case, pDNA and PEI is the main cost driver for upstream viral vector manufacturing. And the use of high-producing suspension cells can significantly reduce the cost in CapEx and OpEx. But the cost of GMP grade PEI and DNA still represent 20% of the price of the batch. And as I said, in fixed bed bioreactor, traditionally we can further optimize the pDNA use and media as well, which can further decrease the cost. So in the same kind of idea to show you how important it is to really optimize as much as possible the upstream and really make sure you focus on that to decrease overall the cost in the end. This is a poster that we published this year at ISCT in collaboration with Vitria, where they performed an assessment of different transaction reagents in the isolate nanobioreactor, and these were hectonine-free T cells that were transfected with one single GFP plasmid with a certain DNA concentration per square centimeter. And all the different conditions, different setups had the same metabolic profile. So when this is the case, we estimate we have a similar cell growth. So in theory, we could expect similar productivity. So what they did here is that they sampled fixed bed strips from the bioreactor from different regions in the fixed bed. They lysed the cells that were on the strips and they measure the fluorescence. What we see on the left graph here is that between PA Pro plus optiferin, which is one of the combinations they tested, and a liposome reagent they compared it to, PA Pro, whatever the position of the strips in the fixed bed, outperforms the liposome. So it's around, let's say, double of the productivity, even a bit more, depending. When they compare the PA Pro, so this is the same data here, with Vectovir AAV and optiferin, which is another reagent from Polyplus that is in theory developed for suspension cells, they see that even if it's an adherent cell, but it's a bioreactor, this transfection reagent for this cell line in this setup outperforms the PI Pro by between 1.5 and 2 times on the productivity. So this outlines the importance of screening different transfection reagents or methods when you're developing your process, because when you do that, you can really find the best setup that by increasing productivity, or maybe it could be also that you don't increase the productivity, but that you find a reagent that maybe uses half or 5 times less than the other one, and that mathematically will decrease your costs. So it's really keeping in mind that when you're developing your process. So when we talk a lot about the upstream, right? So all these things you can do to improve your productivity and therefore reduce the overall cost. But we also need to, it's really important to be mindful that while you're playing with the upstream and increasing the productivity, you will directly have an impact on the downstream cost, the downstream performance, and therefore the cost. And so this needs to be balanced. You need to make sure that while increasing the productivity, you will increase the contaminants, the process-related impurities, and the product-related impurities on the upstream side. You need to make sure that the increase of productivity on the upstream, which will then give you the cost reduction, is higher than the extra cost you are going to get from removing all this new stuff you put in. So in the downstream, to remove the contaminants and all these impurities, you might have to put a larger filter. You might have to put a larger chromatography resin or membrane. And you might even have to add a unit operation in the downstream that you didn't need before. So all this needs to be assessed and thought of at the very start, so that you make sure that it is worth it and that the overall improvement in the upstream will compensate for these maybe small increase you will need to do on the downstream. So as I said, the final lever I wanted to talk to you today as a tool to reduce cost is process identification. This is something that several colleagues of mine are working on. So process identification is kind of a methodology to minimize areas of what we call MUDA or waste. And these are traditionally in either transportation, inventory, all motion. So every time you move something too much, when we wait, when you over-process, so when you do too much that you don't need, when you overproduce, or when you have a defect. And when you're doing process intensification can have benefits in, as I said, in cost, in reducing the footprint, which will link to cost mathematically, in reducing time to market, the time in the facility, the scalability, the ease of use. So I will illustrate this with a case study, which is the COVID-19 vaccine for AstraZeneca. This is a vaccine that was developed by Oxford Biomedica and is based on a genetically engineered chimpanzee adenovirus vector, ChAdOx. The need for this, as you know, with the pandemic, was for large quantities and affordable to supply low and middle income countries, and simplicity and robustness to allow technology transfer to multiple manufacturing sites across the globe. So we worked on this process within our process development lab, and what was done here is that if on the first part of the chart here, you have a traditional adenovirus manufacturing in suspension, where you normally have the bioreactor, a clarification step, and then you have a TFF step before capture by anion exchange. Then you have a final TFF and a point to microfiltration. We developed an intensified manufacturing process where we removed the TFF step here before the anion exchange. By doing that and directly loading the cell supernatant on the anion exchange, we removed one unit operation. We therefore had a shorter production time. We had a similar yield, so it turned out with that process wasn't really bringing a lot, and we had a similar CQA, so within spec. So this is an example, and this has been published in the paper. The second part of process intensification example that was done in this process is around the upstream side, where we worked on the MOI, so on the multiplicity of infection. And the initial process had a high MOI. So on day 1, the vessel was inoculated at half a volume. On day 3, it was fed. On day 4, we had an infection with 1-to-1 high MOI. And on day 6, we had a harvest. What was done here is that we developed a process with a low MOI, where we inoculated the vessel not at half, but at full vessel volume. And we infected a low MOI at day 2. It was that we had then a feed where the virus was able to multiply. And then at day 4, again a feed, and at day 5, we had a harvest. And this allowed for less virus seed needed. And this was reduced by more than 1 order of magnitude, so this is significant. The same production time, similar productivity, and similar CQA. So again, this highlights how through process optimization, so linked to what I was telling you before, you really can decrease the overall costs and sometimes the time of your process. So for this COVID-19 vaccine, there were different drivers. So the first, for example, was the robustness, and for that we used a fully single-use manufacturing platform. For simplicity, as I said, we optimized the infection step with low MOI, and we removed the TFF step between the clarification and anion exchange. So this removes the over-processing MUDA. From time to market and simplicity, we used standardized configuration of single-use assemblies where we could. And also we in order to increase the time to reduce the time to market and to maximize the tech transfer efficiency, to remove waiting, we had a tech transfer support between our global teams. And then for around the cost, the combination of all these solutions above drove the cost down from 1.39 to 0.20 euro per dose. So this is an illustration of how process intensification can help reduce the cost, even for viral vectors, not only for mass. So in conclusion, smart bioprocessing is really important for, but with the use of process intensification, through the use of technologies and performance materials, and through also the use of digital and integration tools. And the key concept around this is really to adapt the product, the process, and the scale, and to predict the performance, the robustness, and the portability. So the goal is to develop the best solution, considering all the different parameters, all the different dimensions, addressing the drivers and the values of the customer or the manufacturer. So with that, I'm happy to take any questions, thoughts. Yeah.
Thank you, Emmanuelle. That was a really interesting presentation. Does anyone have a question? Would they like to unmute? Or even better still, if we can stop the slide share and everyone switch on their cameras, then we can have a good chat. It's a nice small group, so hopefully people are not too shy. Switch my camera on as a start. Does anyone have any comments, questions?
Maybe you don't agree with what I said.
Yeah.
Or you agree.
Exactly. Is anyone out there? You must have covered it so well that everyone's speechless now. Or either that, or they're all on the bus home and they can't speak, because that's sometimes what happens as well. I know that Pei Ching is actually in a car on the way to the hotel, so he couldn't see the visual, but he can, hopefully he might be able to hear us. Does anyone have any comments? Everyone agree with what's been covered?
Linda and also Emmanuelle, so thank you for this session, and my apologies, my side might be a little bit noisy. I'm actually traveling for a customer meeting in the region. So we talked about cost control, right? So definitely, you know, viral vector has a lot of promises to some unmet medical conditions. However, on the other side, I think because of COVID, now we have seen the power of mRNA, and potentially mRNA offers a lot of cost advantages. So really the question to Emmanuelle, you have consulted a lot of customers in Europe for both viral vector and mRNA. How do you actually see the future prospects of these two modalities? Do you think mRNA has a very good chance to overtake viral vectors because of the cost advantages?
I think they will cohabitate, at least for a while, because from what the information I've gathered and from what I've discussed, mRNA yet cannot do all that viral vectors can do. And maybe in the future, it might not even be mRNA that will take over everything. It might be exosomes and targeted depot nanoparticles and things like that. But so far, I think that all the data we have gathered from market research and discussion with customers is that we will have, it's basically we have all these different tools in the toolbox that we just need to figure out which one is the best for what. And certainly mRNA is much cheaper, but it also has, I think, some limitations for certain things that where viral vectors will be required. So I think it's just really figuring out that question about, basically we have a hammer, we have a screwdriver, so which one we use for what? That is really, I think, the big question that the industry will have to figure out in the next months, years. Yeah, that's what I've been seeing.
Thank you, Emmanuelle. Does anyone else have a question? Want to comment?
I saw Pinky Xu put a comment. Thank you, Pinky.
Yeah.
For the feedback.
Yeah, I think Pinky had to leave as well. So, but thank you for the session. Hang on, we have another message. Okay. No, I think we're good. Which means that we'll be able to let you go and carry on with the rest of your day, Emmanuelle. I know it's just starting, right, for you?
Yeah, well, it's lunchtime now. So.
Oh, okay. All right. Oh, I see Julia is on. Hi, Julia.
No, just wanted to say hi to both of you, Linda, of course, and Emmanuelle. It was interesting. Sorry, I missed the first 5 minutes, I think, when you introduced the topic, but it's something that we don't hear that often. So this is really good. And for you to come and speak, and I think your timing wise, you probably, what's the time for you?
Oh, it's 12:15. It's lunchtime.
Oh, all right. Not so bad.
No, it's okay.
It's not sleeping time. Yeah.
No.
Oh, that's nice. Have you been in Singapore? Because you
I
spoke
have
about
actually.
the processes here in Singapore.
No. I have been in Singapore a few years ago for
All right.
a holiday. But
Okay.
no.
Oh, that's nice. All right. So all the best to you.
Thank you. You too. Have a nice end of day.
Thank you,
Yeah.
Julia.
Thanks.
So if there's no other comments or questions, I think we will leave it at that. Want to thank you for sharing your time and your expertise with us in your morning. We hope that we'll be able to welcome you to Singapore in the future. Yeah, and
That'd be nice.
Yeah. And I hope that everyone who's been able to join has been able to get has learned something new from this session, and we look forward to the next one. So with that, have a great evening, everybody in Singapore and Asia, and have a great day, Emmanuelle.
Thank you. You too.
Thank you very much.
Bye-bye.
Bye all.
Thank you.
Bye.
Thanks, Emmanuelle. Thanks, Linda. Bye.
Bye.